For centuries, humanity has grappled with the invisible burdens passed down through generations. Genetic diseases. Conditions that steal lives, limit potential, and leave families in despair. But what if we could rewrite our very biological code? What if we could mend the broken blueprints of life itself? We stand at the precipice of such an era, an age where our understanding of DNA is not just about reading the code, but about editing it. For years, one name dominated this field: CRISPR. A technology so precise, so revolutionary, it earned the nickname molecular scissors. Originating from the ancient defense systems of bacteria, CRISPR-Cas9 allowed scientists to pinpoint a specific error in our DNA, make a precise cut, and sometimes even insert a small, corrected piece. Imagine a single typo in a vast instruction manual for a complex machine. CRISPR offered the ability to find and fix that exact typo. It promised cures for diseases caused by a single, well-defined genetic mistake. The possibilities seemed limitless. Yet, even with its incredible power, classical CRISPR faced limitations. The scissors created a cut, and while our cells tried to mend it, the repair wasn't always perfect. More importantly, many devastating genetic conditions aren't caused by just one simple typo. Take cystic fibrosis, a debilitating lung and digestive disorder. It's caused by problems in a single gene, but there are over 2,000 different ways that gene can be faulty. 2,000 different typos that can lead to the same disease. Treating it with genome editing often requires many mutation-specific approaches. That's labor-intensive, and also intensive from a regulatory standpoint to get all those approaches approved. Imagine trying to develop thousands of different tailored therapies for one disease. The scale of the challenge was immense. Scientists realized they needed a different approach, not just fixing typos, but replacing entire faulty chapters in our genetic book. And now they may have found it. A breakthrough that allows scientists to weave whole functional genes directly into human DNA. This isn't about snipping out a mistake. It's about introducing a brand new working copy of an entire gene into a person's cells. It's like replacing a damaged engine with a completely new one, rather than trying to repair every broken part. This new system, incredibly, also finds its origins in bacteria. But it doesn't use the molecular scissors. Instead, it utilizes what are called CRISPR-associated transposases, or casts. Think of casts as tiny genetic movers, capable of taking large sections of DNA and relocating them, precisely and efficiently, without making a risky cut. This no-cut approach is a game-changer because it bypasses the messy, error-prone repair process that sometimes accompanies traditional gene editing. Casts have been known for a few years, but the challenge was making them work effectively in human cells. Naturally occurring casts were incredibly inefficient, barely able to insert DNA into a tiny fraction of cells. Not enough for a meaningful therapy. So, Dr. Witte and his colleagues embarked on a remarkable journey of accelerated evolution in the lab. They took a promising cast from a bacterium and, using a technique called PACE, phage-assisted continuous evolution, they rapidly engineered it. Imagine compressing years of natural evolution into just hours in the laboratory. It took the scientists over 200 hours in PACE, which corresponds to several hundreds of evolutionary generations. The same process would have taken years with more conventional methods. Through this innovative process, they created something extraordinary: EvoCast. A new, evolved cast that is an astonishing 200 times more efficient at integrating DNA into human cells. This isn't a small step, it's a giant leap. EvoCast has shown incredible promise. It has successfully inserted genes into cells linked to diseases like Fanconi anemia, Rett syndrome, and phenylketonuria, achieving efficiency rates of 12% to 15%, a significant breakthrough for potential treatments. Its capabilities even extend to cancer therapy, showing promise in editing immune cells for CAR T-cell treatments. This opens the door not only to treating diseases inside the body, but also to creating powerful cell-based therapies in the lab. The road from laboratory to clinic is still long, requiring more research to optimize delivery and secure funding. But the potential is undeniable. Evocast offers a cleaner, potentially more effective way to address a vast spectrum of genetic illnesses. And this immense power brings us to the profound ethical questions that inevitably arise when we gain such control over life's fundamental code. The most challenging of these is the concept of designer babies. The term itself often conjures futuristic, even dystopian images of parents selecting traits like eye color, athletic prowess, or even intelligence. While that remains science fiction, the technology that could potentially lead us down that path is advancing. Until now, much of the ethical debate around gene editing focused on somatic cell gene editing. This means editing genes in adult cells like blood cells or liver cells. These changes only affect the individual being treated. They are not passed on to future generations. If a patient with sickle cell anemia receives gene therapy to correct their blood cells, their children would still inherit their original genetic code. But the true ethical frontier, the realm of the designer baby, lies in germline editing. This involves making changes to the DNA in sperm, egg, or early embryo cells. Any edit made here would be woven into the very fabric of that individual, and critically, would be passed down to every future generation. On one hand, the promise is extraordinary. For families haunted by devastating inherited diseases, Huntington's, Tay-Sachs, germline editing could offer a way to permanently eradicate the disease from their family line, ensuring their children and grandchildren are forever free from that genetic burden. A profound hope. But this is where the slippery slope argument becomes starkly real. If we can use germline editing to prevent a severe disease, what about a predisposition to a less severe condition, like a genetic tendency towards obesity or high cholesterol? And where do we draw the line between preventing illness and enhancing human capabilities? With technologies like EvoCast, which can seamlessly insert entire genes, the potential for more complex multi-gene enhancements moves from distant speculation to a more tangible possibility. Imagine, hypothetically, inserting genes associated with enhanced immunity, or even certain cognitive advantages. These are capabilities that blur the lines between curing suffering and optimizing human potential. These are not merely scientific questions. They are deep societal dilemmas. Who determines what constitutes a disease versus a trait which we wish to change? Who will have access to these incredibly expensive, cutting-edge technologies? Will it deepen existing inequalities, creating a genetic elite with enhanced abilities while others are left behind? The implications for social justice and equality are immense. The international scientific community is well aware of these profound ethical considerations. Strict regulations and even moratoriums on germline editing are in place in many countries, precisely because of the irreversible implications for all future generations. But science continues its relentless march forward, and that means we, as a global society, must continuously engage in these crucial conversations. We must collectively define our ethical boundaries. Do we confine these powerful tools solely to eliminating the immense suffering caused by genetic diseases? Or do we, consciously or unconsciously, venture into the realm of human enhancement, with all its unforeseen consequences? EvoCast is a testament to human ingenuity, offering a brighter future for those afflicted by genetic illnesses. It is a beacon of hope. But like all technologies of immense power, it demands profound reflection, robust ethical frameworks, and a vigilant eye on its long-term impact on what it means to be human. The revolution in gene editing is here. The decisions we make today will shape the very fabric of our tomorrow. Let me know in the comments what you think, and while you're at it, hit like and subscribe. It really helps.